the central dogma of biology says that information flow from DNA, it flows to RNA. So, and from RNA, it flows to protein. And I told you that the journey from DNA to RNA is called transcription. And the journey from RNA to protein is called translation. And I don't know if I illustrated that with you. If I did not, I'll still do that when I get there. Now, this here is transcription. This is transcription. And this here is translation. Now, in the actual sense, I remember I wrote it in a very complicated way last week, or more complicated way rather, that shows pre-mRNA and mRNA. So I'll be talking about that since I already showed that last week. All right. So but before transcription can happen, DNA replicates as well. It reproduces itself, which is what we want to look at now, DNA replication. All right. So initial process in DNA. Now, DNA replication actually happened during the interphase stage. Of course, you should still remember that from bar 1. It happens during the interphase stage, and you know interphase have growth phase 1, S phase, which is the synthetic phase, and growth phase 2. The S phase is the synthetic phase, and that is where interphase happens. I mean, that is where DNA replication happens. The synthetic phase means DNA replication, DNA synthesis. All right. Now, whenever DNA will be synthesized, because it is double strand, did I tell you last week about the nature, the structure of DNA, how DNA is anti-parallel, and how they do their DNA, they do their replication in a semi-conservative manner? Did I mention that last week?
Yes.
Yes.
All right, great. I told you that
the
the strand,
the other side is 3.
thank you. They do like one person is sleeping head this way and the other is sleeping head this way. Does that make sense?
Yes.
All right. So we say that they are anti-parallel. And I also told you that the DNA replication is semi-conservative. Does that make sense? Did I say that?
No, you didn't mention that.
Okay. Did I explain to you that every new DNA have half of the old? Did I say that?
Yes.
Okay, good. That's what it means. Semi-conservative means it is half conservative. It means half of the parent is always in the offspring. So every new DNA you see, there is a 50% old, 50% new. Since DNA are double-stranded, so one strand is old, one strand is new. Does that make sense? So there's old strand. Every DNA is never new, 100%. It is 50% new, 50% old. Now, so because of the way they sit, or because of the way these strands are located, that they are anti-parallel. So think about it. If one is 5' to 3', and the other is 3' to 5'. Now, let me tell you, this creates a challenge. Now, the challenge it creates is this. It's like one is head to toe, one is toe to head. So if you are supposed to do something, now, because of that, take note, the enzyme that does DNA polymer, the enzyme that does DNA replication, the enzyme is called DNA polymerase. DNA polymerase means polymerizer, meaning that something that builds larger structure. You know, polymer means larger structure. So an enzyme that helps to join smaller molecules together to become big is called polymerase. And of course, I told you last week that the monomer for DNA is called nucleotide. Do you remember?
Yeah.
And I told you that nucleotides are made up of 3 things. Number 1, nitrogenous base. Number 2, ribose sugar. Number 3, the phosphate group. Does that make sense? Those components makes up the nucleotide. So what DNA polymerase does as an enzyme is simply to join the nucleotides together, like though you are adding blocks together. So when the nucleotides are joined together, they then form DNA. But we are going to be making 2 strands each. So think about it. look at this. Look at what I want to show you here quickly. I said that this strand, these are the parents now, for instance. So one of these will be parent, the other will be parent for the new strand. So let me change the color of this and show you this beautiful stuff. So let's say this is the new DNA we want to make. You know what's going to happen? Now, let me still use red. So imagine they are separating now, and this guy separates, this guy also separates. Because that's what is going to happen first, and I'll show you how it happens. So this is still going to be 5 prime to 3 prime, and it's going to be 3 prime to 5 prime. Those are the old guys. Those are the parents. Then the new will come up. The new here is now going to, this will be a template to form a new one. The new one is going to be 3 prime for this and 5 prime for this. That makes sense? And the new here as well is going to be 5 prime here to 3 prime. Now we have 2 new DNA. This is a new DNA. This is a new DNA strand. 50% old, 50% new. But there's a challenge. You see, this guy will open up. As this guy is opening up so that it can separate, because they were bonded together before. Now, as they are separating so that they can make new DNA, DNA polymerase have a challenge. It can only make DNA in a particular direction. It cannot go, you see this new DNA now, this is in a direction, this another direction. So DNA polymerase cannot make in both directions. DNA polymerase can only make in one direction. So because of that, one will be made in the normal way, while the other will be made in a reverse way. So this one now, DNA polymerase only makes in 5 prime to 3 prime. What does that mean? Let me use another marker. DNA polymerase can only make in 5 prime to 3 prime. What that means is this. There must always be something that is existing with 5 prime to 3 prime direction before DNA polymerase can do work. So I say that DNA polymerase is not a starter. It cannot start something. Something else needs to start it and create an empty 5 prime to 3 prime direction, such that DNA polymerase can now come and add to the 3 prime side. So this side is where DNA polymerase can come and add nucleotides to. So if this side does not exist, DNA polymerase cannot do anything because it doesn't work from the scratch. It's not a starter. So what is that can help DNA polymerase to make the nucleotides to stay? It is called RNA primer. So the first thing that will be made, anytime you need to make a DNA, the first thing you must make first is called RNA primer. And of course now, ladies will understand it better now when you say primer. What do you use primer for when you are doing makeup? Talk to me.
For the start.
Eh?
Starter.
To set the face.
As a starter, right? To set the face. Now, what would be the advantage of using primer?
It helps
The
the
makeup
foundation,
film.
the makeup to last longer. Something like that.
Stay.
Yeah.
Yeah.
Does that make sense? So if you don't use primer, it will smear quickly, right?
Yes.
Thank you. But if you use a primer, it will make the foundation to stay well and it will last long. That's the same thing that RNA primer does for DNA. If there is no primer, you cannot have a DNA. The DNA will not stay. I have to start using mine. Okay, that's cool. It will make it last longer. All right. All right. DNA. So this is so before DNA polymerase has to come, there's an enzyme that helps to make primer. The enzyme that makes primer is called primase. Take note of that. So primase is the one that will make RNA primer. Take note, the primer is an RNA, just like the primer you use is not foundation. It is not a foundation. It is not the deal. It's just a primer. So the primer is an RNA. It is not a DNA. So the primer needs to be made first. Why? Because DNA polymerase is not a starter. It can only polymerize, meaning that it can only add. So if nothing is existing, it cannot add, because so it cannot add. All right. So RNA primer needs to be made first. So when the primer is made, the primer will be made in this direction, 5' to 3', such that DNA polymerase can now come to add in this direction. So that means the strand that is 5' to 3' is called the leading strand. Is the strand you can make straight. So this strand now, this strand here is going to be the strand you can make straight, because you see what's going to happen? As these two strands open, a primer will be made here. As this RNA primer is made here, 3' end is open. DNA polymerase just come and continue the whole process and be adding it. So we say that this strand is the leading strand, which is the 5' to 3' strand. We say it's a leading strand. We say it's a continuous strand, because it's just DNA polymerase will just continue straight and make it straight. No gap, no break, because it is in its direction. But look at the unfortunate one, or the one that will be delayed. Now, this guy, we call it lagging strand. Lagging because it's going to be delayed or discontinuous. Why do we say that? Look at it. This 3' to 5' cannot be made easily. And of course, you can be thinking why already. The reason is this. This strand is in the 3' to 5', and DNA polymerase only can only add onto 3 sides. So look at what is going to happen. So whenever this strand opens up, and I will talk about the enzyme that opens it up. When it opens, as it's opening, DNA polymerase is going to come here. When it opens, DNA polymerase cannot start at the edge, just like this one rather. Sorry, the DNA polymerase can just start here and be doing the combination straight. But here, DNA polymerase cannot be doing it, because DNA polymerase can only add in this direction. It cannot add here. So now, what's going to happen is DNA polymerase is going to wait and let this strand open up a little bit. When it opens up, then DNA polymerase is going to come here, like this now, come here, and synthesize this in the direction 5' to 3'. And then it's now going to add the nucleotide to this area, like this. It's not going to add it. So what do you notice? There's a primer here. It's going to wait again and let it unwind again. It's going to come here again and make a primer somewhere here. And then the 3' is open here, right? So it's going to make it again, add it again. So now you're going to have primer in between. Does that make sense to you? Let me know if it makes sense to you.
Yes.
Okay. I'm not getting responses now. Does that mean you are not getting it? Let me know so that I will know what to do.
Can I ask a quick question?
Please go ahead. Yes.
So when we're referring to the leading strand and the lagging strand, those are not the new strands being created, but the original ones before?
No, we are talking of the new strand being made.
Okay. Thank
The
you. That's
way
why I was
they are
confused.
made. Yes, the way they are made. So the leading strand, see, the old one is what is opening up. And as it opens, it serves as a template for the new one. So the one that will be made, this one here now is the leading strand, is the one that will be made in the 5' to 3' direction, which is the way DNA polymerase can also make strand. DNA polymerase can only add to the 3' side. So now this one will just make one primer. You see, one primer, 5', the primer is 5' to 3'. And then DNA polymerase can just add, because the 3' end is open. It will just add in a continuous manner. We call that leading strand. But here, because of this direction of the new strand, DNA polymerase cannot make it like it's making this. But does it mean it will stick for long? Okay. Let me see if you're asking a question. I'm not sure if I got that, but I saw the other part. Now, so DNA polymerase will now wait, since it can be made in a strand. Okay, don't worry, I'll show you another picture where I will explain it better. Let's go. All right. So key thing is this: DNA polymerase can only add on the open 3' end. So take note of that just in case this is confusing to you. DNA polymerase can only add to the side 3' that is open. That's where it can add to. But before that can happen, that means there has to be something that have that direction, and it's always an RNA. So that tells you that you can make RNA from the scratch, but you cannot make DNA from the scratch. Go ahead, Florence.
Excuse me. So that means
Sure.
it can be add from the 5 to the 3, but it cannot be add from the 3 to the 5.
No, look at it. So let's say you have 5' to 3'. This is a primer, right? It is 5', right? And 3' here. Does that make sense? If RNA primer want to add, if DNA primer want to add, DNA primer can only add to this side. It cannot add to this side. Does that make sense? So if you now bring this here, that's what I'm saying here, that DNA polymerase can add to this side. So whenever you now want to make this strand that is going to be the challenging strand, it will not, let me clean this and then do it all over to show you. So whenever you want to make this strand now, now look at this. Whenever you now want to make this strand that is 3' to 5', DNA polymerase will wait and let the strand opens well. When it's open, it's now going to come here and secrete RNA primer maybe somewhere right here. So this RNA primer is going to still be like 5' to 3'. Then DNA polymerase will make the new DNA to this area. Can it add to this side? No. So what will it do? It will wait again and let it open very well. Then it's going to come here and make another primer to this place. 5' to 3'. And then DNA polymerase is going to come. Primase is the one coming to make primer, primase. So DNA polymerase will now come, then make DNA to this area. So to make it up to here, then it will stop. The same thing is going to come here, make another primer, then make DNA and stop. Does that make sense? So now what it means is this: we will end up now having DNA fragments and not continuous. Number 2, we have many primers in between the DNA that we will need to fix because you must remove it. If you don't remove it, then that means you have an hybrid of DNA and RNA, but we don't want to have that. We are going to later need to remove it, which I'll show you. Does it make sense to you now? If it makes sense to you, please let me see your response on the chat. Right? Let me keep saying it if it makes sense to you. Right? And if you have questions, please ask. Okay, great. Thank you. If you have questions, please ask. All right? Now let's go to the next thing. It's still the same thing. I'm still going to emphasize it. So that is it. The primer is connecting the, okay, good. What the primer does is, primer is an RNA. We call it primer, but it's actually it's an RNA, but we call it primer. The reason we call it primer is because it has to be made as a primer before the foundation, which is the DNA itself, will be added. The primer is just there as a connector. But we will need to remove the primer later from the largest strand because we are going to have multiple primer, right? So we have to remove it because we want to make DNA. We don't want to make DNA and RNA. Because think about it, A must be pairing with C. A must be pairing with T. G must be pairing with C. So if you have an RNA, which usually can have U, then there's going to be a problem in the DNA. So we are not going to have RNA and DNA at the long run. I'm going to explain how that will be done. So now I deliberately explained it with that difficult screen that I did with my image, just to make you see it. If you can understand that, then it will make sense to you when I'm explaining with the real image now. So this is what happened. This is a DNA strand that is still coiled on each other. Please bear with me. This is a DNA strand that is still coiled on each other. And then Before DNA can be replicated, number one thing that will happen is that it needs to unwind. If it does not unwind, there's no way you are going to make another DNA strand from each of them. So number one thing is that it will unwind. What is the enzyme that makes it to unwind? The enzyme that makes it to unwind is called DNA helicase. Thank you. It's called helicase. You can see it on the screen. Helicase. Thank you. Now, helicase does the unwinding. However, there's another enzyme that we call topoisomerase. That topoisomerase, it helps to relieve the strength of the bond. That's its job. It helps to relieve the strength. Helicase now comes to unwind. So in quotes, you can say topoisomerase will weaken it. And then helicase comes to do the unwinding. So this is helicase here, unwinding. When helicase unwinds, it creates a fork. We call it replication fork. But see, think about it. Helicase, a 2 DNA strand has been winded or has been wound to each other together. Helicase is now coming to unwind them. Does it not have possibility of winding back on each other, just like your twist? When you do your twist and you are trying to take it out, does it not have tendency to coil back together? Now, it has tendency to prevent it from coiling back on each other. There is a protein. It's actually called SSBP. SSBP, it means single-stranded binding protein. It means single-stranded binding protein. This protein will bind on the strand that has been separated at the replication fork to prevent it from coiling back. So as the strands are being separated, single-stranded binding protein steps on it to prevent it from coiling back. Now, the next thing is this. After that, DNA polymerase. So now, how will that happen? Before that will happen, primer had to come first. So now let me show it to you here first. Primer is going to come for this strand that is 3' to 5'. Don't forget, this is the template strand. These are the parent strand. So if I want to illustrate it from here now, the strand, again, which strand is leading strand from the previous slide? Which strand is leading strand? Yeah, very good. The strand that is 5' to 3'. That's what I want to hear. Does that make sense? The strand that will be 5' to 3', which is the continuous strand, is the leading strand. So looking at these templates now, don't forget, we don't judge leading strand by the parent. We use the parent to judge it, but the leading strand is the new strand, not the parent strand. All right? So this strand here is the one, the new strand that will be made here has to be made in the strand. Let me use a different marker, a different color. The new strand that will be made here now will be made as 5' will be here, and then 3' will be here. So that means this is not going to be my leading strand. The strand that will be made here will be made in the 3'. Don't forget, the partners have to be anti-parallel. So you can't have 3' and 3' here. No. All right. So this strand here will be 5' to 3'. So in this strand now is my leading strand. What does that mean? It means this will be the strand that will be made just with one primer and it will be continuous. Because you know why? Oh, sorry. I was messing it up. I was messing it up. Permit me to clean this. Okay, all the instructions I made are correct anyway. It's the leading and lagging strand that I messed up. So look at something here. Oops. Sorry. let me show you this to illustrate it so that will save me time of annotation as well. So again, the parent strands are called parent strands. They are not called leading or lagging strand. But this will be my leading strand here. This is why it is. Let me show it to you. This is the 3'. This is the 5'. I told you that DNA polymerase can only add in the 3' side. Don't forget that 3' side. So the 5' here. So I wish maybe I'll play a video for you. So look at it. This is 5'. This side means that this side is the 3'. So since this side is 3', DNA polymerase can make this. So as the fork is opening, DNA polymerase can also be making it alongside. So it makes it alongside with the fork. So imagine now, if the fork is opening, it keeps opening, DNA polymerase just keep going. Nothing is stopping it. It keeps going until they get to the end, and then we name that end 3' because it can only add to the 3'. Don't forget that. Just keep it somewhere in your notes. DNA polymerase can add to 3'. So we end up producing the strand that will end in 3'. So as the fork is opening, then it will be making it like that. It just keeps going because the 3' end is open. But on this side, the 3' end is not going to be open because the strand here is the 5'. And then the new strand we want to make here will be the one that will be 3' end here. So imagine now if this ends as 3'. So that means DNA polymerase is supposed to be adding from this way, this way, this way. And that will not happen because the fork is opening this way, in this direction. Fork is opening in this direction. This strand is supposed to be made in this direction. So that's the problem. So now because the direction of the strand and the direction of the fork is different, so they will be made in fragments. So see what happened here. Primer is going to come, secrete a primer first here. When you see, okay, so I will just talk about this side. It secretes a primer here. Our DNA polymerase now comes in and build the DNA here. Comes again, secrete a primer here. DNA polymerase then come and make DNA strand here. Again, it's going to come, secrete a primer here. DNA polymerase will come and make the primer in this direction because they are following the reverse order. Now, what is going to happen afterwards to the primer? That is actually what I'm going to talk about now. But before then, each primer, we call it Okazaki fragment. Each segment of DNA. Because they are made in small, small fragments, so we call it Okazaki fragment. Okazaki is not anything. It's just the name of the person that make it. It's not the name of the person that discovered it. All right? So Okazaki does not mean anything. It's the name of the person. So we call it Okazaki fragment. So this is Okazaki fragment here. Another Okazaki fragment here. Another one is being made here already. So what's going to happen afterwards? Do we want to have DNA that have RNA in between them? No. You notice the RNA has been removed from here. So what will remove the RNA? 2 enzymes. Number 1 enzyme is called DNA ligase. Number 2 enzyme is called DNA polymerase 1. So what is going to happen? The DNA this guy that makes the whole thing is called DNA polymerase 3. You can see that. DNA polymerase 3. So now let me explain something to you. DNA polymerase 3 and DNA polymerase 1. What is their difference? DNA polymerase 3, I'll just use 3 to show it. I said that 3 is a big guy. Because it's a big guy, it doesn't go in between. So DNA polymerase 3 cannot come in between here now. DNA polymerase 3 cannot come in between here. It cannot come in between here. It cannot come in between here. It can only stay at the 3' end. So we say that DNA polymerase 3 only have, we call it, let me type it so that it can be better. We say that DNA polymerase 3 only have, oh, cool. We say that it has exonuclease activity. I don't know, I have this privilege before. We say it has exonuclease activity. See that? We say DNA polymerase 3 only have exonucleic activity. Exo, meaning that it can only do anything from the outside. It cannot go in between. So DNA polymerase 3 can never come in between. It can't come here now, like where you see DNA polymerase 1. DNA polymerase 3 cannot come in between there. It can only come, you see, when you make a primer here now, there is a lot of space. DNA polymerase 3 can come here and then make it and then get out. But the gap, the location where you have the primer, DNA polymerase 3 cannot just come here. But DNA polymerase 1 is small. Because it's small, it's tiny. It's able to do exonuclease activity. So DNA polymerase 3, DNA polymerase 1 can do, number one, it can do exonuclease activity as well. It's able to do exonuclease just like DNA polymerase 1, number one. Additionally, it's also able to do what we call endonuclease activity as well. Call it endo, meaning that it can go in between. Now, somebody should be thinking in your mind now that what happens when there is error in DNA? Interestingly, DNA polymerase 3 can do this. Look at this. DNA polymerase 3 have something that we call proofreading activity. So we say that it has proofreading activity. What does that mean? It means that as it's working, it can proofread. So as it's adding the nucleotide, it can proofread, but it has a challenge. So let's say that it's adding this way now, and it notices an error immediately, it can fix it before making another one. So if it's supposed to add, let's say it's supposed to add A, C, T, A, then it mistakenly add C when it wants to add G. It can correct it immediately and then put the G there. So you see, even this thing that I'm using to write now, once I make a mistake, I can't come there to correct it and make it, and no. But before, you see, look at this. Look at this. I can type something now. If I make a mistake, I can correct immediately. But when I take my mouse from there, I can't come back to correct that mistake again. That's the way DNA polymerase 3 is. If it's making a mistake, it can correct the mistake immediately. You see, I want to delete 1G, I can delete it. But if I take my mouse off and come back, I can't delete again. Can't edit again. So DNA polymerase 3 is exactly like this. I'm happy I'm using this now. It can edit as it's going. So we say it has proofreading activities, but it cannot come back to fix an error. So if it makes an error now, it can't come back to fix an error later in between. So it is DNA polymerase 1 that is tiny that can come back to fix an error in between. So this is it. So what is going to happen to the RNA primer? You see, this guy, RNA primer, will need to be removed. But what will remove it? Number one. All right, let me close this now. Number one, you see, ligase, number one, DNA polymerase 1 will come in to remove the RNA. You see, DNA polymerase 1 is here now. It has removed the RNA. This is RNA, the primer is going now. What is now going to happen? Ligase is going to come and fill the space with DNA nucleotide. So who does the job? DNA polymerase 1 comes in, cuts the primer, remove it. DNA ligase comes in. We say that with the word we actually use is that we say DNA ligase will seal the nick, meaning that you seal the space there. So DNA ligase will seal the nick. DNA polymerase 1 will cut off the primer. Does that make sense?
Yes.
All right. So that is the way it works. I'm going to clean this now. Again, take note, DNA polymerase 3 have exonuclease activity and proofreading activity, whereas DNA polymerase 1 have exonuclease activity and endonuclease activity. It's able to go in between. Like here now, DNA polymerase 3 cannot come here now. to remove light primer. It can't. But DNA polymerase 1 can do that. All right, let's go. So this is 756, right? I think, let me show you this video, even though you are not going to hear the volume. I know you are not going to hear the sound, but I want you to see the what do you call this thing now? Is it the animation now? How it's playing out? I want to see how the lagging and leading strand is playing out. All right, does that make sense? All right, so that's it. Now, what is interesting about, so this is something I want you to be picturing based on what I've taught you. If you remember, I told you the difference between bacterial chromosome and eukaryotic chromosome. Prokaryotic chromosomes, do you still remember the shape of the chromosome? What does prokaryotic chromosome look like? Thank you. It is circular and it is just one. But in eukaryotic organism, it is linear, multiple millions of linear DNA. So because bacterial chromosome is one, it makes it faster to replicate their chromosome because it is one and it is circular, right? So once it opens up, it just goes in the same direction and that's it. So it can be fast. So that makes gene production, gene expression to be faster in bacteria than in humans or than in eukaryotic organisms. So that's key thing I wanted to take note. So you see, their DNA replication for bacteria is bidirectional. They use gyrase and topoisomerase to remove supercoils. Supercoils mean that the coils, all right? Now, DNA is methylated as well, and I'll talk about that later. It helps with control of genetic expression, initiation of DNA replication, protection against viral infection, repair of DNA. Now, what does the methylation mean? Methyl, you know, I mean, some of you that did chemistry a little bit, you still know what methyl is. Methyl is CH3. Methyl is CH3. All right, that's methyl. We call it methyl group. Now, the DNA, they usually methylate it. Do you know why it's methylated? Now, this is it. The purpose of methylation, it helps to identify the DNA strands. So it also helps sort that if there's an error, you know the DNA strand to repair, if it is the new one or the old one. So imagine now, let's say that there's an error and both new DNA and old DNA are there. Now, how will the polymerase or ligase, how will they know which one to repair? The only way to know which one to repair is if they are able to know the difference between the strand. Because how will they know the new one and the old strand? And if there was a mistake on the new strand, how will they know if they should correct it? I will give you an illustration now. Let me see this. Excuse me, please. Okay, so let me draw this. I think I can use this illustration here. Now, this will just be best. So let's say I have a DNA strand that is A, T, C, I mean G, C, T, A. All right, this is my DNA strand. This is the parent. Now, if this is the new strand, don't forget I'm the one using color to differentiate my strands. So let's say this is my new strand, and it is going to be T, A. You know, it's pairing because you remember I told you last week, it's called Shagaf theory. All right, A pairs with G, A pairs with T, G pairs with C. So this will be C, this will be G, this will be A, this will be T. So these are the strands. These are the new strand now, or this is the new strand now, the old strand, the new strand. Now let's say during replication, let's say during replication, DNA polymerase mistakenly put C here instead of T. By the time they need to repair this, how will they know which one to repair? Because an option is this: if it is about making it accurate, which is what the job of the repair mechanism is to make sure the gene, the nucleotides are matching. So they can come here and put a G here instead of, and put a G here and delete the A. Now, in the real sense, in terms of matching, it is matching. But was that the original? No, the original is supposed to be AT. GC will be matching and correct, but it will be a mutation because there will be a problem. Because it was not originally GC, it was originally AT. But GC is matching, but that is actually a problem. So how will the repair system know which one to repair and know that you don't come to repair that A is correct to be here and that what needs to be repaired is G is here, so that you turn this to A to T instead of turning this to C to G? The way to know the repair is that the parent strand are usually methylated. So the parent strand will carry a methyl group. Now, the methyl group helps to identify that this is the parent, and that is the reason that you will know that this is the parent. It means that that is not the strand to manipulate. It is the new strand that you should correct. And so that is why we put that there, that it helps to protect against viral infection, helps to repair DNA majorly. All right, that's that. All right, so this is the way DNA replicates in prokaryotic cells. You can see that is the origin, and then it just open up from the origin and goes in both directions. So we say that DNA replication happens in both directions, bidirectional, all right, in DNA in prokaryotic cell, right? And so that continues. Now at the end of the day, you're going to have two strands, all right? Now, DNA replication of eukaryotic cell, for instance, I told you it's not going to happen it's not going to be as fast as prokaryotic cell, all right? It's similar to bacterial replication. Some differences occur. Of course, there are differences. Prokaryotic have shorter Okazaki fragments. The Okazaki fragment in eukaryotics, rather, I think is about maybe 50 and not up to 1,000, right? So they use this for DNA polymerase. So there's DNA polymerase 1, polymerase 2, polymerase 3, polymerase 4, all right? They have thousands of replication origin. Eukaryotic cell have thousands of replication origin, whereas prokaryotic only have one replication origin. Plant and animal cells methylate only cytosine bases, right? It's only cytosine bases that plant and animal cell methylate, that they put methyl on to identify their group. All right, let's go. Now, what is the difference between genotype and phenotype? I believe I started to talk about it last week when I talk about the central dogma of biology. Do you remember I told you that, of course, what's the difference between genotype and phenotype? Who wants to talk to me? DNA. What's the difference between phenotype and genotype? I'm not seeing anything on the chat, and I'm not hearing any voice. Phenotype is the gene that is expressed. Genotype is what your genes are, Genotype. Okay, cool. Thank you. So genotype is your physical look. Is a physical. Phenotype is physical rather. Genotype is gene. It's from the word gene. So think about this as gene now. DNA is your genotype. So here is genotype. This is what determines your genotype. Your protein is actually what determines your phenotype. Take note of that if you've not thought about it that way before. Because this is it. When your parents, oops, I'm sorry, I won't type it again. Okay. Just take note that phenotype is the gene that produces protein is phenotype. Is what determines your phenotype. The gene that is inside of you but did not produce protein, we call it genotype. I mean, it means that would not help. The gene that is inside of you generally is your genotype. But the one that produces your protein, the one that produces protein is the one that determines your phenotype. So you got a gene from your father and your mother. You have one gene from father, one gene from your mother. Your daddy is 6 feet. Your mother is 5 feet. You got the gene for 6 feet. You also got the gene for 5 feet. But fortunately for you, it's the gene for 5 feet that is expressed in you. You are 5 feet tall. You are not 6 feet tall. But you have a gene for 6 feet inside of you. When you give birth to your first child, your second child ended up becoming 6 feet tall. And then yourself and your husband is 5 feet. And then they are wondering, how did your second child is 6 feet? It is because there's a gene inside of you that you got from your 6 feet father, but that gene refused to produce protein inside of you because the gene that produces protein is what determines your phenotype. So the reason I have this skin color is because the gene that produces protein produces the protein that determines the phenotype. So if there's no protein production, the gene becomes recessive gene. So the gene that did not produce protein is the recessive gene. The gene that produced protein is the dominant gene. Does that make sense? And the one that produces protein is the one that determines your phenotype. All right? So that is the difference between phenotype and genotype. So I'll stop there today because so that I can talk about this. Of course, I've been talking about central dogma of biology since. Right? That's the central dogma of biology that I've been talking about. So I'll stop here today so that we will start from here next week. On Thursday when we meet.